Good morning, biochemist, and welcome to what really is the last part of our lipids series. Okay, so now I'm going to introduce you to a new functional group, and that is the phosphoester or phosphate monoester. So a phosphoester or a phosphate monoester is the addition product between an alcohol and a phosphate. So a regular ester is going to be the addition product between an alcohol and a carboxylic acid, and this reaction is going to look very similar to that. It's another double replacement reaction where we have water as one of the products. So in other words, it's a dehydration reaction or a condensation reaction. So if we can make a phosphate monoester, do you think we can make a phosphate diester? Well, yes, we can. All we do is react the phosphate with another alcohol and we can form a phosphate diester. And this process is going to produce two water molecules instead of just one. So why is this important? Why are we talking about this in a lipids lecture? Well, the reason is cell membranes. So phospholipids, which contain a phosphodiester linkage, are the major component of biological membranes. And there's two types that we're going to talk about. We're going to talk about glycerophospholipids and phosphosphingolipids. So glycerophospholipids look like this right here. So you're going to have two fatty acid residues, a glycerol residue, a phosphate residue, and an alcohol residue. So I want you to pause the video and see if you can tell me what type of linkages we find in a glycerophospholipid. So these fatty acid to glycerol linkages here, those are going to be esters because we have a carboxylic acid and we have an alcohol. Now this right here, where we have a phosphate attached to two separate alcohol residues, that's going to be a phosphate diester linkage. So what are these good for? We already mentioned that they're the major component of biological membranes. They're also going to be a minor component of myelin sheaths that insulate our nerve cells. They're a component of lipoproteins, which we talked about in our last lecture. And they're good emulsifying agents, and if you don't remember anything about emulsifying agents, be sure to look over section 6.10. Okay, so our next group of lipids are going to be phosphosphingolipids. So a phosphosphingolipid looks like this right here, where we have a sphingosine attached to a fatty acid, a phosphate, and an alcohol. So what is a sphingosine? So a sphingosine is this molecule right here. This is 2-amino 4-trans octadecene 1,3-diol. It's a mouthful. So this is an unsaturated 18-carbon amino alcohol. So these have a very similar structure to glycerophospholipids. So our sphingosine is essentially providing the same role that the glycerol and one of our fatty acid residues did in a glycerophospholipid. So what are these guys used for? Well, this image on the right is a bit of a hint. They are also known as sphingomyelin because they are the major component of myelin sheaths, and they're also a minor component of our plasma membranes. So go ahead and pause the video and see if you can determine what type of linkages we have in a phosphosphingolipid. So the sphingosine and the fatty acid are linked by an amide linkage. So we have the amino group on our sphingosine forming an amide with the carboxylic acid head on our fatty acid. And so then we have a phosphate diester linkage between our alcohol, our phosphate, and the hydroxy group at position 1 on our sphingosine. So our next group are glycolipids. So a glycolipid is a sphingolipid that does not contain a phosphate group. And so the sphingosine is attached to a sugar instead of a phosphate and an alcohol. And we can subdivide these glycolipids into two types based on the type of sugar they're attached to. So a cerebroside has a monosaccharide attached and a ganglioside has an oligosaccharide attached. So what are glycolipids used for? So there's three different functions of glycolipids I want you guys to know. So the first one I want you to know is cellular recognition. So these sugar residues on the glycolipid sticking off the cell act as a binding site. So these can bind to complementary compounds on neighboring cells, drugs, hormones, or even viruses. So they're also good at stabilizing cell membranes, especially nerve cells. And they're used in conjunction with glycoproteins on red blood cells to determine our blood types. Okay, so now that you guys have a little bit of an introduction to the lipids that make up our cell membranes, let's talk about the membrane itself. And we're going to do that with what's called the fluid mosaic model. So this is a mobile view of the cell membrane. And this is the idea that our cell membrane is constantly in flux and it's extraordinarily fluid. And what's going to make it flow so well, what's going to make it so fluid, is the bilayer of phospholipids. And those are going to be highly unsaturated, and that's going to prevent packing. But we don't want this bilayer to be too fluid. So that's going to be the major purpose of cholesterol in biological membranes. They're going to add a little bit of rigidity or structural integrity. So one of the fascinating features of biological membranes are that they are selectively permeable. So this means that only some things are allowed across. And there's 3 processes that are going to govern this. So the first process is simple diffusion. So diffusion is going to be the movement of substances across a semi-permeable membrane from areas of high concentration to low concentration. So water is able to do this freely. Small non-polar molecules, and uncharged lipids. So how do we get other things across a cell membrane? So what if we need to transport an ion, or we need to transport a large biomolecule like a protein? Well, this is going to require either facilitated diffusion or active transport. So we already talked about simple diffusion. That's just movement from high concentration to low concentration, and that's what you see in figure A here. So facilitated diffusion is going to be similar. This is going to be movement from high concentration to low concentration. So it is thermodynamically favorable, but it's involving polar molecules that otherwise could not get across the cell membrane. So how do we get them across? We use what we call a transport protein. And we're going to talk more about that when we get into protein function after our next exam. And so our last process also involves a transport protein. And this is called active transport. And this is what we use when we need to move something across a membrane against the concentration gradient. So when we have to move something from areas of low concentration to high concentration. And now that is not thermodynamically favorable. And so we have to couple that with something that is super favorable. That way the entire coupled process is thermodynamically favorable. And that super favorable process we're going to couple it with is the hydrolysis of ATP. Alright guys, that's it for lipids. I will see you next time when we start my favorite subject, proteins.